Quanta Bio. Ignite a spark. Fuel more insights. The Quanta Bio SparQ mRNA-Seq kit enables efficient preparation of stranded mRNA-Seq libraries for Illumina NGS systems in less than 5 hours. This streamlined workflow reduces hands-on time and delivers reliable mRNA-Seq libraries from as little as 5 nanograms and up to 1 microgram of high-quality total RNA. There are 2 boxes for this kit. Box 1 is to be stored between 2 and 8 degrees Celsius. Box 2 is to be stored between -25 and -15 degrees Celsius. The SparQ mRNA-Seq kit is intended for molecular biology applications. This product is not intended for the diagnosis, prevention, or treatment of a disease. This video shows the process of preparing 8 libraries using master mixes at each step. The volumes presented are on a per-reaction basis. Before you begin, prepare the reagents that are not included in the kit. These reagents include the 10 millimolar tris buffer, the nuclease-free water, 80% freshly prepared ethanol, and the adapters. Mix 300 microliters of the SparQ Pure Mag beads with 6 microliters of the bead booster for each library preparation. This mixture generates enough beads to take 1 sample through the entire library preparation process and should be kept at room temperature during the preparation process. At each step, master mixes can be prepared for multiple reactions and stored on ice until use. This step isolates the mRNA for consistent exonic read coverage across varying RNA input amounts and reduced bias in GC-rich transcripts. The required reagents for this step have yellow caps. Start by pre-programming the thermal cycler with the heated lid set to 105 degrees Celsius. Start the cycling program to preheat the block to 70 degrees Celsius and then pause while getting reaction tubes ready. This cycling program will be used twice within this step. Bring the oligo DT beads, the binding buffer, the suspension buffer, and the wash buffer to room temperature prior to use. Thaw the RNA, the RNase inhibitor with the yellow cap, and the mRNA frag prime buffer with the blue cap on ice. Make the RNA binding master mix by combining 38 microliters of nuclease-free water, 71 microliters of binding buffer, and 1 microliter of RNase inhibitor for a total volume of 110 microliters. Mix well. Store the RNA binding master mix on ice. Thoroughly vortex the oligo DT beads and transfer 35 microliters to a new thin-walled PCR tube for each reaction. Add 110 microliters of the RNA binding master mix to each tube. Add 5 microliters of the RNA. Mix by pipetting and briefly centrifuge. The reaction volume should now total 150 microliters. Mix well. And resume the cycling program. After incubation, briefly centrifuge. Place on magnet until it is clear. Then discard the supernatant. Add 180 microliters of the wash buffer. Mix by pipetting. And then place on magnet. Discard the supernatant. Add 50 microliters of the suspension buffer. Vortex and centrifuge, and incubate by repeating the cycling program. Add 50 microliters of the binding buffer. Mix well. And incubate for 10 minutes at room temperature. Place it on the magnet until it is clear, and then discard the supernatant. Add 180 microliters of the wash buffer. Mix by pipetting. Place on the magnet until it is clear, and then discard the supernatant. This is the purified mRNA. Remove the tube from the magnet with the cap closed, and then place and keep it on ice. In this step, the RNA is fragmented and primed for cDNA synthesis. The required reagent for this step has a blue cap. Pre-program the thermal cycler with the heated lid set to 105 degrees Celsius. Start the cycling program to preheat the block to 94 degrees Celsius and pause. Prepare the frag and prime master mix by combining 4 microliters of the mRNA frag prime buffer and 12 microliters of the nuclease-free water per reaction for a total volume of 16 microliters. Centrifuge. Add the 16 microliters per reaction to each sample with beads. Mix well. Briefly centrifuge the reactions and resume the cycling program. After fragmentation, briefly centrifuge the tube. Place it on the magnet until clear, and then transfer the supernatant to a new tube. The first-strand cDNA synthesis occurs in the same reaction tube from the previous RNA fragmentation and priming step.
Remove the first strand enzyme mix with the brown cap from the freezer just before use and thaw on ice. Pre-program the thermal cycler with the heated lid set to 75 degrees Celsius. Start the cycling program to preset the block to 25 degrees Celsius and pause.
Combine 4 microliters of the first-strand enzyme mix to the 16 microliters of fragmented and primed mRNA for a total reaction volume of 20 microliters.
Note that the first-strand enzyme mix is viscous, and care should be taken when pipetting. Mix well and resume the cycling program. After first-strand synthesis, briefly centrifuge the reaction and place on ice. Proceed immediately to the second-strand synthesis and end polishing. The second-strand synthesis, strand marking, and end polishing occurs in a single step in the same reaction tube. This step generates the directionality of the end libraries. The required reagents for this step have green caps. Thaw the second-strand buffer on ice. Remove the second-strand enzyme mix from the freezer just before use. Pre-program the thermal cycler with the heated lid set to 75 degrees Celsius. Start the cycling program to preset the block to 16 degrees Celsius and pause. Prepare the second-strand synthesis master mix by combining 20 microliters of the second-strand buffer and 10 microliters of the second-strand enzyme mix, for a total of 30 microliters. Add 30 microliters of the second-strand synthesis master mix to 20 microliters of the first-strand synthesis product for a total reaction volume of 50 microliters. Mix well and resume the cycling program.
Briefly centrifuge and proceed to the post-second strand cleanup.
Libraries need to be cleaned up prior to adapter ligation. The samples will be highly viscous prior to cleanup because of the sequential enzymatic reactions.
Ensure that the SparQ Pure Mag beads and bead booster mixture has been kept at room temperature for at least 20 minutes before use. Add 90 microliters of the beads to the second strand synthesis product.
Incubate the mixture for 5 minutes at room temperature.
Pellet the beads on a magnetic stand until clear.
Carefully remove and discard the supernatant. Keeping the tube on the magnetic stand, add 200 microliters of freshly prepared 80% ethanol to wash the beads. Pellet the beads on the magnetic stand for 30 seconds and then carefully remove and discard the supernatant. Repeat the wash one more time and discard the supernatant. Air dry the beads on the magnetic stand for 3 to 5 minutes, or until the beads no longer appear shiny. Take care not to over-dry the beads. Remove the tube from the magnetic stand and resuspend the dried beads by adding 69 microliters of 10 millimolar tris buffer and mix well by pipetting up and down. Incubate the beads at room temperature for 2 to 3 minutes.
Pellet the beads on a magnetic stand until clear. Carefully transfer 67 microliters of the supernatant into new thin-walled PCR tubes
and proceed to the adapter ligation step. Alternatively, the sample can be stored at 4 degrees Celsius for up to 24 hours. The protocol has been optimized with SparQ UDI adapters, but is also compatible with any full-length Y-shaped adapters.
An alternative protocol for use with stubby adapters is available separately.
The adapters may need to be diluted using the UDI dilution buffer before use.
Please refer to the product manual for optimal adapter ligation efficiency and recommended dilution strategy.
The required reagents for this step have orange caps. Thaw the rapid ligation buffer on ice. Remove the T4 DNA ligase from the freezer just before use. Pre-program the thermal cycler with the heated lid off. Start the cycling program to preset the block to 20 degrees Celsius and pause. Combine 20 microliters of the rapid ligation buffer and 10 microliters of the T4 DNA ligase for a total volume of 30 microliters of the ligation master mix.
Add 3 microliters of the diluted UDI to each sample from the cleanup step.
Transfer 30 microliters of the ligation master mix to each tube containing the sample and the adapter for a total reaction volume of 100 microliters. Mix well and resume the cycling program. Briefly centrifuge and proceed to the post-ligation cleanup. Please refer to the post-second strand synthesis cleanup step for the detailed protocol. The bead input and resuspension volumes differ at each step. For this step, add 70 microliters of the SparQ Pure Mag beads slurry to the adapter ligation product. Wash the beads twice, each time with 200 microliters of freshly prepared 80% ethanol. After air drying the beads, resuspend the beads in 25 microliters of 10 millimolar tris buffer. Carefully transfer 23.5 microliters of the supernatant to a new thin-walled PCR tube and proceed to the library amplification step. Alternatively, the sample can be stored at 4 degrees Celsius for up to 24 hours. Thaw the Hi-Fi Plus master mix and the primer mix with white caps on ice. Pre-program the thermal cycler with the heated lid set to 105 degrees Celsius. Take note of the recommended number of PCR cycles at step 3 of the cycling program. Start the cycling program to preheat the block to 37 degrees Celsius and pause. Prepare the PCR master mix by combining 25 microliters of the Hi-Fi Plus master mix and 1.5 microliters of the primer mix for a total volume of 26.5 microliters. Add it to the 23.5 microliters of the purified adapter ligated DNA for a total reaction volume of 50 microliters. Mix well and resume the cycling program. Briefly centrifuge and proceed to the post-amplification cleanup step. Alternatively, the sample can be stored at 4 degrees Celsius for up to 24 hours. Please refer to the post-second strand synthesis cleanup step for detailed protocol. The bead input and resuspension volumes differ at this step. It is important to note that there are 2 bead cleanups within this step. Perform the first bead cleanup by adding 45 microliters of the SparQ Pure Mag beads to the library amplification product. Wash the beads twice, each time with 200 microliters of freshly prepared 80% ethanol. After air drying the beads, resuspend the beads in 52 microliters of the 10 millimolar tris buffer. Carefully transfer 50 microliters of the supernatant into a new tube. Repeat the bead cleanup step by adding 45 microliters of the SparQ Pure Mag beads. When resuspending the dried beads for the second time,
Add 22 microliters of the 10 millimolar tris buffer. Then, carefully transfer 20 microliters of the supernatant into a new tube. The sample can be stored at 4 degrees Celsius for up to 24 hours, or at -20 degrees Celsius for longer-term storage. Proceed to the library QC step. One option is the qPCR-based quantification with the SparQ Universal Library Quant Kit. The libraries are now ready to be sequenced. Quanta Bio. Ignite a spark. Fuel more insights.